So I'm gonna start with just saying we have Meagan from the USGS office in Woods Hole today, and I'll ask Meagan to sort of present herself, how degrees and where did you do your PhD and all that for people who may not know you. And then we'll start from
Awesome.
there.
Yep. Thanks. Yeah, so I'm a research scientist at USGS in Woods Hole, so I'm part of the Coastal and Marine program. I did my master's with Adina Paitan out at Stanford and looked at carbon cycling in mangroves many decades ago, and then moved to work at Woods Hole Oceanographic, and where I ended up actually doing my PhD with Matt Charette. And there my focus was on trace metal and nutrient and radionuclide cycling in subterranean estuaries, and then subsequent groundwater discharge fluxes to the coast. I then moved over to USGS as a postdoc, switched back to carbon, but I've been mixing subsurface cycling and sediments and pore waters and fluxes to the ocean across coastal ecosystems. My focus the last couple of years has been pretty strongly on carbon cycling and looking particularly about the impact of blue carbon environments. So this was fun to go back to some of the trace metal work that I've been doing with Joe. So it's exciting to kind of get back a little bit to things that I had started before, but I just want to say this work is collaborative with Joe Tamborski, Aleck Wang, Matt Charette, and Kevin Kroger. Just a second. There we go. So I'll start with this cartoon of the subterranean estuary and think about, okay, why do we measure things in the subsurface, and then what are the implications for the coastal ocean? And so this system basically shows that there's a large infiltration of terrestrial water inland. That water enters the groundwater table and then flows towards the coast due to a hydraulic gradient. So our water levels are higher inland, lower at the sea. However, during that flow, that water can accumulate a variety of different chemical constituents, either from direct addition, such as nitrogen in this picture, that's one of the examples, or through chemical reactions, either weathering reactions or other things that are going on during transport. So that water chemistry changes as a function of residence time in the aquifer, as well as land use and land characteristic practices above that. However, when it gets to the coast, it meets and mixes with recharging marine water. And so that method of recharge can be tidal pumping, or it can be kind of longer scale sea level changes that induce water flow, marine water intrusion and flow into the coast. And that creates a mixing zone between fresh and salty water, and further chemical reactions occur. It's quite a dynamic zone, and it can set the chemistry for the fluxes that ultimately discharge into the ocean. And those fluxes are important because that's what the estuaries and coastal ocean experience, and can have negative or just some impact on those ecosystems, as well as influencing global cycles of various constituents, from trace metals to radionuclides to nutrients. And so this is kind of the context and the classic example of a subterranean estuary. And when we think about, okay, what does that look like at the coast? This is a classic example of a salinity mixing zone. So we have that fresh groundwater coming in. So what you're actually looking at is a 2D cross section of the coast. So if you were standing on the beach, this would be under you. You wouldn't be able to see it. And the kind of classic image of terrestrial groundwater coming in, marine groundwater intruding from those kind of processes I discussed, tidal pumping, as well as kind of the position of the sea versus the groundwater. And this is that mixing zone. So, that region where it's actually quite small spatially, and you can kind of compare it to the salinity mixing zone in a surface estuary, although it's, like I said, much tighter constraints in space than you would expect in a surface estuary, where the salt wedge could go on for several kilometers, for example. And so how geochemists tend to get at flux is by looking at the concentration in the different end members that they're interested in, and then the flow of water associated with that particular body of water. Those are not totally easy to get at because you have to understand something about where you think the flow is occurring and then what its chemical makeup is. But these are regions of really high gradients, and so sometimes there's a large uncertainty. So this is the classical view as a subterranean estuary. What I'm going to talk about today is Woquoit Bay, where we alter our subterranean estuary a teeny bit. But first, so there's been a lot of work in Woquoit. It's a great place to do groundwater research because starting with Ivan Valiella and Matt Charette, folks have been looking at groundwater in the system for decades. So it's got a rich data set to work on. And so just to show you where the groundwater is coming out, so Woquoit Bay is on the left. It's a fairly urbanized estuary system. You can see housing along the edges, but there's also a large portion of it that is protected through the Woquoit Bay National Estuarine Research Reserve. So at the head of the bay up here, there is the research reserve. And then this portion here is owned as well as a portion over here and is protected. And so that's not developed at all. We have all of our sewage right now is on septics. So there's lots of nitrogen coming into the system. And in terms of how much groundwater is flowing, this is thermal image and indicates cold water. And our cold water is our groundwater discharge. And so you can see cold water plumes coming here at the head of the bay. And then if you look along the edges, there's additional groundwater seepage. And so there's groundwater seepage pretty much along the edges of Woquoit Bay, including over here, which is where, so this is salt marsh. And so there's groundwater inputs under our salt marshes as well. And even where we have surface water expressions that we call rivers, they're actually incisions into the groundwater lens. And so our freshwater inputs are almost nearly all groundwater, just because we sit on a glacial moraine and there's not a lot of large areas to have accumulation of surface water flow. So our surface water is groundwater. And then so previous work in Woquoit Bay on groundwater has basically, this is just a briefly shown that this zone is a source for barium, iron, and manganese, but is a uranium sink. So this is that sandy subterranean estuary work that's gone on. And so what I'll show today is work that we've done out at Sagelot Pond. So this is a salt marsh in Woquoit Bay. It's in one of the sub estuary or like the sub bays of Woquoit Bay. And what you're seeing here is our sampling scheme. And so what makes this region unique compared to some of the other subterranean estuaries or coastal aquifers that you might think of is that there's this wedge of peat that sits atop a sandy coastal aquifer. And at Woquoit, the peat wedge starts at about 2 meters deep and then grades to shallower and shallower as you go inland. So it's a fringing marsh. The oldest peat is around 2,000 years out here at the bottom of the peat complex. And then this marsh is actively migrating inland as sea level rise occurs. And so what we ended up doing was sampling transects from 2014, 2015, and 2016 through both the peat and then the sandy subterranean aquifer below it. We have end member characterization of the surface waters here. This work is in collaboration with large time series efforts to understand what carbon exports have been from salt marshes. And so that work has been published, but this look at the subsurface work has not yet. We also have end member characterization of in a groundwater well that's right here. It's kind of right at the terrestrial boundary within the trees, but just right at the tree boundary. And so we can look at this cross section and look at what is the cycling of trace metals as well as carbon. So that's what I'm going to be showing you today. I've divided the rest of the samples into regions that are indicative of this sandy coastal aquifer underneath the peat, as well as samples that are taken within the peat. And so you'll see that repeatedly through different slides that I show you today. And I'll note that this sampling density of 8 profiles, we only did in 2014. And then for 2015 and 2016, when we did seasonal measurements of Porewater, we had just these sites that are in the round circles. So there were 3 profiles through the subterranean estuary there. What did we measure? Everything. So this is us sampling. You can see here our samples for radium. We actually have 2 depths going at the same time. We have 2 pumps going. We were very ambitious sampling for DOC, DIC, and methane. I'm not going to show you any methane data today. It's mostly DIC and DOC, as well as nutrients. No nutrient data today. We'll show you trace metal data, but no radium data. So this is a super rich data set that is telling us a lot about what's going on within this region. So what does our salinity structure look like? And so if you remember back to the one I showed you earlier, there was a freshwater wedge sitting atop marine groundwater intrusion. So this is a little bit different than that. So first to walk you through this is our depth profile. So it goes down to 2 meters. And then this is our transect starting landward and going seaward. So this stops at the creek edge, about 1 meter from the creek or the edge of the estuary. And as you can see, our peat depth increases as we go more seaward. So this line is that peat sand interface, we think about it. And here we have marine water sitting atop the kind of like the fresh intrusion. And that is because of the daily overtopping of the salt marsh, which leads to input of marine water directly into the peat. That water then infiltrates and flows through the peat and then discharges into creeks and along the edge of the marsh. And it typically discharges kind of at the elevation of the low tide and higher through time. And so this is our salinity structure. And what does it then look like if we look at other parameters? So here's the pH. I'm going to, just hide some of you so I can see my notes. Here's the pH of the water, and you'll notice that it's quite low, particularly within the freshwater portion of the subterranean estuary. But it's always below 7. So it's acidic, which is what we expect. The main reactions here in the peat are sulfate reduction of organic matter, and then subsequent reactions of minerals and other things in these low pH environments. It's highly reducing. So this is ORP. None of these values are above 0. So the entire system is reducing, but it's highly reducing within the peat. So those values, are negative 400. It's a very qualitative measure. We don't use this to indicate exactly what reaction is going on, but we know it's sulfate reduction. However, there is some input of oxygen with the terrestrial groundwater. So you can see kind of an oxygen plume that inputs at the landward edge and then attenuates with that flow and as various reactions use up that oxygen as it goes through the subterranean estuary. So what are our end members here now when we talk about this? And so I'm going to show you a variety of plots that are looking at different trace metal behavior, and then we'll get at different fluxes that we think are associated with these different processes. And so the inputs of water to the system are obviously the overtopping from the bay, and then the fresh groundwater input as it comes in. And those meet and mix and will discharge with unique chemistries that are different from what the groundwater looks like coming in and what the bay water looks like coming in. And so we're gonna now assess, through mixing plots, largely starting at our kind of like property salinity plots to look at, is there conservative mixing, is there removal or addition? So we're starting with iron. And the first thing I'll note here is I've put in conservative mixing lines between the groundwater end member and then the estuary end member in all of these. I'm not showing you the actual estuary data, so we're not digging deep into, how does the surface concentration vary through time, because that's a whole other talk. But we're looking at, okay, within the subterranean estuary, what's going on. The sandy portion here is going to be in our green squares, and then the peat portion is going to be in our blue triangles. And I've attempted to keep that color scheme throughout most of the talk. And so you'll see here whoops, there we go. The iron is pretty high in our fresh groundwater. And then we have a region of the sandy subterranean estuary, kind of at the oxidized edge of the peat, where there is quite a bit of iron release. And then we have kind of this deep iron removal sink here, and that is associated with the portion of the subterranean estuary we believe is forming iron sulfur minerals, whether they're exactly pyrite or not, you'll see a little bit later in the talk, as well as some iron release actually going on in the marsh. If you look at this in terms of the redox, so this is now plotted versus oxidative reductive potential versus salinity, you'll see that there's a really sharp redox boundary that's controlling kind of the dissolved iron. And so that formation probably of iron minerals at that redox boundary is essentially serving as a sink. However, there's enough iron release in other portions, and the iron concentrations are still actually higher than either the input from, the seawater input. And so this, while a lot of the iron brought into the system is trapped, there's still some iron export, which you'll see shortly. Uranium. So as expected in a highly reducing environment, there's a large amount of uranium removal. So pretty much all the samples fall below with large removal in both the peat and the sand subterranean estuary. However, if you look at uranium versus iron, they're largely decoupled. So ignore the groundwater samples for now. Just look at, okay, where is uranium? Where do we see some uranium? And that's in typically the marsh sediments. And where do we see iron release? And that's typically in the sand subterranean estuary. And that just has to do with what different, I think, minerals and phases are in solution at the different pHs and the reducing at those two different sites. So the uranium and iron are both being removed, but their removal is decoupled between the peat and the sand subterranean estuary here. And so this is one example of where kind of our classic understanding of how these systems operate separately, we can operate together in a dynamic way, because you can have peat that isn't atop a sand subterranean estuary, and you can have a sand subterranean estuary not associated with this peat, reducing peat cap essentially. So manganese is ubiquitous throughout. There's release everywhere, and that's probably because as you'll see in a minute from some data that Joe shared with me, the manganese minerals are there. It doesn't favor manganese mineral formation, whether it's manganese oxides or other manganese carbonate or other manganese minerals. And so this is a region for a large release of manganese, likely due to weathering of a variety of clay and other minerals in the low pH environment. And then barium is also released, and this is a combination of both, the ionic strength increase. So barium typically desorbs and peaks between 5 and 15 for the salinity, but we also are having weathering that's releasing barium. So we're seeing the input of both of those influences to enhance the barium concentration of the Porewater and the groundwater here. And so this data is from Joe. He's been working with me on this, and we're looking at doing some freak modeling of the subterranean estuary with a particular focus on the trace metals. And so we're below barite saturation, or barite is in equilibrium at the salinity zone, but it's below saturation in the peat, and that's because there's a massive removal of sulfate in that region. And then those iron minerals, goethite and hematite, are super saturated in the marsh peat. I don't think we pulled out exactly the kind of like that region where it looks like there's large iron removal with compared to the peat, because in some of the portions of the subterranean estuary, we're actually seeing what it looks like iron release. And so I think that we'll dig into that a little bit more. And then obviously manganese minerals are undersaturated, which is allowing kind of manganese to be in solution. And a lot of this is these kind of clay bearing fine grain clays, and they're sites of really active cycling, especially when we get into this low pH reducing environment. And so it's essentially kind of like enhancing weathering at a rapid rate, especially because of the flow of water through the system is removing the products rapidly. Oh, sorry. I didn't realize I added this slide, Joe. I'm going to go to this one. So this, Joe is collaborating with somebody who's looking at X-ray diffraction. So this is the mineralogy. And it's been really interesting to get some confirmation of some of the modeling results to look at, okay, what minerals are present, and look at, you know, the clays, because those are what we think are actively cycling and producing the enhanced mineral dissolved flow that we see. And so this is still something we're working on and looking at, and how to combine this exactly with the freak results. But there's definitely large weathering products of some of these quartz and other minerals. And we're looking at to see, like, how does this compare between the peat and the subterranean estuary. So now I'm going to go into some of the trace metal fluxes, and we've scaled this across all of Waquoit Bay. And this is then leaning on some of the previous work that's been done. So we're dividing it into 3 different sources. So there's terrestrial groundwater, and by terrestrial groundwater, I mean at the low salinity groundwater. There's indications that that water does discharge directly into the bay. Not all of it is mixed with marine groundwater. So we have this terrestrial groundwater component. The flux rates for that are based on hydraulic models that were from a paper I published in 2013. And then the chemical end members for that are based on work that Matt Charette did earlier. And then we have marine circulated bay water. And those rates are based both on rates that we looked at with hydrologic models, as well as radium and other kind of isotopic tracer methods. And so that rate of water flow is a combination of those different approaches. With this kind of the chemical signature, again, is a combination of both the marine subterranean estuary that we measured in this study site at Albert St. Gilles Pond. So here, as well as combining the data from the head of the bay. And they're actually pretty similar for a lot of the trace metals we're looking at. And then this Porewater exchange is what goes on as the water infiltrates, and then exits the marsh peat. And what I did is scaled it to the entire bay. And so thinking about this on the kind of the scale of the bay, we took the entire area of salt marsh peat that occurs adjacent to the bay, as well as where we know groundwater discharge is occurring. And so this was I think the scale that I felt confident that we could compare at and try to understand what the relative importance is of these different processes. And just to go a little bit in depth on how do we know what the Porewater exchange is. So this is data that was collected from the radium kind of balance method. I won't go into how we do that, but we took radium isotopes at different time series, and we know kind of what the export is, and we know what the groundwater concentration is. So we were able to get both a submarine groundwater discharge and a Porewater exchange. And basically to look at those is they vary a lot through time, and year to year, as well as season to season. But I used an average across this entire time, which is kind of at the level that we're looking at this. And so groundwater discharge is about 1/10 of what the Porewater exchange is. And I want to just say that that is counterintuitive to some folks, because if you think about how water is moving through peat and sand, it definitely moves through sand a lot easier. So at this site, like I said, we have a really deep knowledge of the hydrology as well as the geochemistry. You know, the movement of water based on slug tests through the sand is 32 to 150 meters per day. Whereas an infiltrometer test, which isn't exactly apples to apples, but is a measure that we can compare, is much lower than that, you know, a half to 1.7 meters per day. And so while the hydraulic conductivity is much less in the peat, we actually have a lot more water flowing. And I think that's due to that daily kind of overtopping and recharge of what can flow out, as opposed to the driving force for some of the subterranean, or like the submarine groundwater discharge. So that's different than the Porewater exchange. It just doesn't have as much volume to get out in this region. And so calculating all of that, so I took the end member concentrations times the water fluxes for all of those, and then partitioned the flux into the groundwater. So that's the terrestrial groundwater, a marsh, so that's the Porewater exchange, and then a sand subterranean estuary. So that's that portion of the aquifer, that's that marine recirculated and mixed water. So those are the three distinct sources we used. In terms of units, iron and manganese are in kilomoles per kilometer squared per year, and barium and uranium are 3 orders of magnitude lower than that in moles per kilometer per year. So we'll see here that except for iron, marsh-derived fluxes really dominate. And by flux, I also mean this uranium sink here. Over the sand subterranean estuary in Waquoit Bay. And, you know, and area wise, there's more sand subterranean estuary in Waquoit than there is marsh land in Waquoit. And about half of the sand subterranean estuary in Waquoit is covered under by a marsh. And so that's kind of the ratio there. Although that is decreased through time. And so it's actually entirely possible that some of the sand subterranean estuary now actually has marsh on top of it, but we don't know because it's developed. And I didn't include developed areas in that. But it's entirely possible somebody's front lawn is a former marsh. And so you can see like the kind of the rates are fairly high, but the behavior of a uranium sink is what we would expect in a marsh environment. I didn't scale these beyond Waquoit Bay because actually the area of marsh globally is really not well known. There's kind of wildly different estimates, but it's a huge number once you scale it globally. But just to say that these areas are important if we're trying to think about, okay, what are the global potential sources on like, you know, oceanic scale trace metal fluxes, we would want to consider, particularly for, you know, iron, uranium, and barium. So now I'm going to swing us over to carbon. And you know, the kind of the sulfate reduction that is the dominant form of organic matter decomposition is really setting up the geochemistry of the subterranean estuary to result in all those trace metal fluxes I just talked about. And at this site, we've thrown a variety of different methods. This is a table from Joe's paper, but the MLR tidal model, some of, you know, that was developed, and the Chanos data was developed by Aleck and then our group at USGS. And so, you know, there's 4 different ways we've kind of hit at a carbon export from this site, which is pretty unique and really exciting because all of these methods are very independent of each other. And the fluxes of carbon are large. And so this is a focus on DIC right now. And that flux is about 4 times greater than the burial of soil organic carbon, which is about 150 grams per meter per squared. So, and then unpublished, the DOC flux is about a third of the DIC flux. Kevin's working on that right now. And so that means that by far the largest fate of carbon in these systems is that the plant's uptake is brought into the system and then exported as lateral flux. And so there's thoughts about like how do we think about that flux and what it means in terms of blue carbon, but it's a large flux. And so some of the work that I've been doing is looking at in the pore waters, okay, what is the production rates and what is the source and age of that? And so this is an alkalinity versus DIC plot. I've shown the 1-to-1 line. And I just want to show here that that all that sulfate reduction is producing DIC in excess of alkalinity. So this is likely due to those coupled reactions that we talked about, because this isn't exactly the ratio you would expect with sulfate reduction. So this DIC enhancement it suggests that there's an alkalinity sink, which I'll show in just a sec. And then this is a plot of DOC versus DIC. And the DOC in the terrestrial groundwater is screamingly high, but there's good evidence that it's attenuated very, very rapidly. And that's along actually the fresh groundwater flow path. By the time it meets and mixes down here with the peat, it's already quite low, and then it further attenuates. The groundwater DOC or the Porewater DOC and the sand subterranean estuary DOC actually doesn't vary very much throughout that region. It's about 1,000 to 2,000 micromolar. So there's the rapid DOC attenuation of terrestrial DOC, but then cycling. And so where is the DIC here? As expected, it's enhanced, and this is going back to our cross section, it's enhanced in the peat. So we're producing a ton of DIC there. But interestingly, if you look at alkalinity, you'll see that there's some decoupling of the alkalinity production. And that's largely due to this sink here in the deeper peat. And so that's kind of likely due to the mineral reactions that are going on there, kind of enhancing alkalinity. It's being produced, it's just now being stored in the peat. So going out, okay, so we have this, we know DIC is actively being produced and cycling out into, to support these large exports of DIC. So what is its source? And there's hundreds more samples than this that I've run DIC carbon isotopes on. I'm just showing you a subset because these are the subset I actually have carbon-14 measurements. So I can talk to you about source and age. And so my end member mixing model is not based solely on the samples you see here, but on additional samples. And so this is just a plot of DIC concentration versus salinity. The mixing model is a function of salinity, DIC concentration, and DIC isotopes. So I haven't shown you that in this parameter space, but we're able to partition and figure out the fraction of marsh carbon that is derived from these various sources. And when we look at that, there we go. The Porewater carbon is about 90% marsh derived on average, and that's what you would expect. You saw those large increases up to like 10 millimolar, which is an increase, of 8 millimolar over surface water. So there's just very large inputs of marsh derived with a carbon isotopic ratio of about -12 to -13. About 10% of the surface water, so this is when you measure it in the surface water, is marsh derived. And groundwater contributions to kind of like both surface and pore water are on the order of about 13%. So these are the kind of the averages. So, the pore water system is marsh like we expect. And then there is input of that marsh derived carbon into the kind of like this large background marine pool that floods in. The flooding water is full salinity, so it has about 2 millimolar DIC, and so you're inputting a small amount of DIC into that carbon. So how old is it? So we took a subset of samples and measured carbon-14 on both DIC and DOC, Porewater as well as groundwater. And I'll say measuring C14 on sulfidic samples was a very big challenge. So we worked with no SAMs, didn't get it right, and then developed a methods that we think these are right. And so I'll just say that this is age in years. These black dots are the ages of the sediment organic carbon at these elevations. And so it's elevation, but you can also think of it as depth. It's just to put samples that were collected at different parts of the marsh kind of on the same datum. And so you can see, as expected, soil organic carbon age increases with depth. I'm sorry, I keep changing the slide. What really surprised me was that Porewater DIC doesn't. It is modern, and we're going to zoom on this in just a sec. But there are several places where Porewater DOC actually does have a higher age than modern. And then these two groundwater ones, these are collected at that groundwater well, and they're also modern. And so that suggests that there's a decoupling between the soil organic carbon source at depth and then the dissolved carbon that's at that same depth. So this is when we now blow up into the kind of like, okay, the past decade. And so this is age actually since it was collected. So you'll see as I kind of expected, there was a gradient as with age increase in the groundwater. So the sample that's kind of right at the surface, and it's not at zero, so it's not at the surface, it's just at this elevation, it's 15 centimeters below the sediment surface. You can see there is an increase in age with depth, but for the Porewater DIC, which is in the triangles, it's about all the same age. Doesn't really vary in terms of its carbon-14 content and its del-13C content. It looks about the same. And then with the DOC, we had some ages that were coming in at, essentially, it wasn't aged at all. And so, what this is starting to suggest to us is that there's the source of the carbon is likely plant-mediated respiration. That carbon gets entrained into the pore water and then circulates through the aquifer and discharges. It doesn't necessarily interact with the soil organic carbon pool, although there is some evidence from the DOC that there might be some interaction. But the DIC, which is the main respiration product and the main carbon export, does not appear to be aging as it transits the subterranean estuary. And so just looking at, okay, some of the surface water carbon. So we did over one tidal cycle. We have some C14 measurements for both DIC and DOC. Again, the DOC is the only samples that have expressed an older age than modern. And I'll just note that, one of these is on a rising tide and one of these is on a dropping tide. And so I don't know, there's no hydrologic rhyme or reason to why these ones might be older than the other ones. And so, I can't fully explain why we sometimes have modern DOC and sometimes have older DOC, unless it's just not fully and well mixed at that particular site. But if you zoom into just the DIC age, you can see again, everything's modern. These two squares are Vineyard Sound. So this is like all offshore water, so not collected in the estuary, but these are off, like kind of more what I would expect the Atlantic to look like. And these are modern, but these were actually duplicates collected at the same time. And so they kind of like span the entire range of the DIC age that we see on the surface waters. And so that just argues that, all of this carbon is relatively young. We're not seeing export of old carbon. So if we bring this back to this carbon cycling graph that we think about. I guess I would argue that this arrow here from carbon burial to carbon export, we're not really focused, that's not a strong a supplier of carbon that's being exported. The Porewater age on average for DIC is 8 years. It's older for DOC, but that's bimodal. There were 2 old samples, and then all the rest that we collected, of course, our N for DOC was only 6. Were young and modern. But in the creek waters, you know, again, we had a higher number of DIC than we did of DOC samples. DOC, I'll just say, also was really hard to measure comparatively for the C14. And so it just largely suggests that we're not decomposing our soil organic pool, and causing the carbon export is due to modern plant inputs, not to relict carbon being lost, which is good in terms of we're not burning through our marsh right now. And thinking about what does it mean for carbon benefits and blue carbon. If you're burning through relict carbon, that's really different than if you're taking up modern carbon. So take homes from that whirlwind trip through all this this subterranean estuary with peat on it, is that this combination of peat on top of sand really produces kind of some unique geochemistry, especially when the interactions are. And the flow rates, especially when you think of, most folks think of the low hydraulic conductivity in salt marshes as impeding flow, but because they are regularly flooded and then the water levels drop, you can induce quite a bit of flow through these regions. And that is really just amplifying the geochemical cycling, and particularly because they have such fine-grained sediment compared to some of the sandy subterranean estuaries. Some of the weathering reactions are favored and can occur more rapidly, especially as the products are removed. The carbon cycling is really rapid and it's young. And we're exporting the vast majority of the carbon that's brought into the system via photosynthesis, via lateral exchange. Burial is not the main fate. Local burial is not the main fate. And our DIC exports were always modern, while DOC varied. And I'm not sure what's driving the DOC age. And so that's what I have for you today. I would be happy to take any questions or discuss any of that.
Thank you, Meagan.
There's everybody.
Here's everybody. All right. So questions, guys.
Ideas. Hey, Kevin.
Yeah. Hey, Kevin, go ahead.
Hi, Meagan. Really interesting. It's fun to see the work that I've been on the edge of, part of, but so it looks, so with the peat being so much lower hydraulic conductivity, do you have a sense of, is it acting as a confining bed that's getting thicker and thicker as you get towards the shore? Is there some effect there on the flow?
Yeah, so I went into this thinking that the peat was going to be acting as a cap, and I think that there is indication that there's preferential flow of that groundwater under the peat. But I think that that's still the volume of water that's flowing through there is much less. And we've hit that several different ways, and it's still much less than the water that's circulating through the peat. I think our peat is fairly, you saw the infiltrameter rates. They're pretty high for peat, right? There's some peat where you put an infiltrameter on it, and I don't think you can actually measure it because I think ET might be faster. And so I think our peat is fairly conductive. And so do I think, in terms of it acting as a cap, I think there is some entrainment of fresh water at the base, and they're cycling there. But I do think it's mostly then encouraging that fresh water to discharge. And I don't know about offshore. The bay is filled with mucky sediment. And so I don't know where exactly it's expressing, but we are seeing evidence of fresh groundwater in the surface water. So I think it's discharging somewhere where we can still see it in the tidal water. How's that for a geochemist's explanation?
Okay, no, yeah, thanks. It's really interesting, the patterns, and it looked like where the peat got really thin, there might be more of a connection with the sandy aquifer just based on the salinity distribution.
Yeah, and I definitely agree with that. I mean, and where it's thin, there's a higher sand content, because that's like marsh. That marsh is like just grown. Some of it is like a decade old. And I think that because it does, I think more water is entrained that's marine there, but it rapidly then flows. And so it's even possible that that's sliding down that peat layer, if that makes sense, right?
Okay.
Because it would be saltier.
Sorry. We have more questions. So I first saw a hand from Songji, if I pronounce the name properly. Can you unmute and ask your questions?
Yeah, that's right. Yeah, thank you. Thank you, Meagan. So it's a lot of information here, so I'm not sure if I missed it. Or so when you are saying lateral flux of, for example, DIC, are you referring the DIC from the marsh Porewater to surface water, or like from the area close to the marsh into the estuarine? So I like to.
Yeah, lateral. When I say lateral flux, I mean the kind of the dissolved transport via tidal exchange, and that one of the inputs to those tides is Porewater drainage. And so the Porewater then enters the tidal channels, likely at low tide, and then exports out as the tide leaves. Does that help?
Oh, okay. Yes. Okay, then I have a follow-up question. So I noticed that there were huge difference in terms of DIC flux using different method. So one of the method you used is the radiant and thorin ratio method. So I'm wondering, like, how many cores did you collect to get the ratio of, I know it's a lot of work to get that flux rate, but I'm just wondering how many cores were you able to do?
Yeah, so Joe Tamborski did this. He was up here as a postdoc with Matt Charette. And so we actually kind of joined forces. I had done a lot of the radium sampling previously. So he did 2 to 3 cores per season. And he, I don't have the figure here, but he measured those over multiple seasons over 2 years.
Okay, and then so you use the average of those values. Okay.
Yeah, and it was, you're right, it was quite different.
Yeah.
Is Joe still here? If he can answer more.
Joe had to leave at 9:30 because he had to proctor an exam.
Okay. So
I think he already have that. Did he have that paper published?
Yeah,
I remember seeing it in the conference a few years ago, but.
Yep. So this is from his 2021 paper. Oh, sorry, I don't know why. Oh my goodness. It's from his 2021 paper, and that's an LNO.
Okay, all right, I can check that out.
Bye, Kevin.
We have one more question by Ruby.
Hi, you answered part of it right now. I was curious about what season the data had been taken to understand seasonal dependence, and also had a question on how does whether you're at low tide or high tide affect things in terms of which way the flux is going, and is there significant tidal variation?
Yeah, so in terms of sampling seasons, so the Porewater and surface water sampling occurred from 2014 through 2019. And across spring, summer, and fall seasons, we don't really have winter Porewater data. However, the MLR surface water concentration model was deployed during the winter because that is based on modeled concentration data and then high resolution and high frequency flux data. And so, yes, this is about a 1 meter tidal amplitude. So it is microtidal, but it's not a lot. And we do see greater fluxes at actually the greatest carbon fluxes occur right at both into the marsh and out of the marsh, with coincident with the highest velocities, and our highest water velocities occur at the top of our tide. So right before the flood ebb change is where our velocities peak for our particular tidal system. And so that's where the greatest fluxes occur, but the greatest concentrations occur, like of pore water amended elements, occur in low tide, but our actual water velocities during those periods are very low. And then there was one other part of the question that I forgot. What was it? Oh, and then so pore water sampling, I don't show it, but the pore water sampling at low and high tide, for anything that's deeper than 5 centimeters is the same concentration. So the temporal, or the scale of temporal change is not the tidal time scale for those samples. And most of our samples are focused below 10 centimeters in the marsh. And then seasonal variability. So, we designed these samplings to really get at pore water seasonal variability, because we see a huge amount of seasonal variability in the export in the summer, but we're just not seeing seasonal variability in the pore water. And so trying to kind of explain that has been a bit of a mystery, and I'm still not there. I don't understand how we can maintain such high concentrations in the pore water, but without a coincident flux, because the flux, all the work that we've done suggests that water is fluxing through the marsh, except for when it's frozen. So our marshes freeze in January and February, and then we don't get any exchange then. It just basically is a sheet of ice. The water comes in, the water goes out, and we don't do very much, except for the very deep groundwater might be flowing, but the pore water is just stuck.
Thank you.
All right. I have a question. I was intrigued by the ages of the DOC and DIC. I have to say I'm not super surprised, because we work on similar peat in Mobile Bay in Alabama, and we dated 15 samples and all came very young, and we sort of gave up on that. I was like, wow, we were expecting to see some very old carbon, and we're seeing something like 60s and 70s. So I was wondering, in regards with the export of carbon, so obviously this is very young carbon, do you think there's something to do that with the stability of the different types of organic carbon? Have you done carbon DOM quality analysis? And look at yeah, I was hoping.
I haven't done any of that. Yeah, no, that's a great question. First, I think you should publish those data, because it's important to know that it's modern carbon, right? Because there's been a couple of publications saying, oh, this is, these exports are only being supported by respiration of ancient carbon, right? And so I think it's important to show the opposite. We haven't done any analysis of the DOM at all, just to show, why is it all so young, and why is, there's that kind of like the age-old argument, right? Is the preservation of this organic pool because of the environment, or because of the characteristics of the DOM or the DOC, or whatever you want to think about it? And I was shocked at how much DIC production there is. It has to be just chewing on labile.
Yes, right.
It's crazy.
I was disappointed that it's so young, and I was like, I'm, I was really doubting the results and all that. And that's why, we are preparing this paper for publication, but I wasn't planning to put the ages in there because I wasn't sure that's correct. But now I'm more confident.
Oh, definitely. I think you should put them in. I mean, it's a really interesting question about like, okay, so that means that injection of DOM from the plants is essentially, supporting respiration as it flows through the marsh. And so it has to be, less recalcitrant than obviously that carbon that's just sitting there. When we split open these marsh sediments, like most marsh sediments, I mean, the leaves are identifiable and they're 2,000 years old. And so I don't think it's not because that material can't be broken down. I think there's just such a ready source of material that is preferable.
Mm-hmm.
Yeah.
Actually, our solid material, we picked up some pieces, woody pieces, and they came young too. And so I was, I was surprised. My original hypothesis, this is some old buried, carbon from the last sea level rise and whatever and all that, but it didn't work that. We are doing actually DOM characterization, and Hannah is doing this for us, and she was in the audience, but she just left. I was hoping to pick up our mind, but super interesting how, things are overlapping and, learning from each other, and it's giving more confidence about results and whatnot.
Yeah. No, did you do the DOC and the DIC pools?
DIC.
Yeah, the DOC, man, that was a struggle.
Yeah, we sent it to Uthal, actually, my previous student, and he graduated, of course, didn't write a paper, left, and now I'm buried with work to do, writing, but yeah. Do we guys have more questions for Meagan? Super interesting stuff. So thank you so much, Meagan. And I hope to see everybody soon, sometimes in person, in conferences. Thank you so much. See you guys.
Right, thank you.
Bye.
Bye.